Hello, everyone. Welcome back to my YouTube channel. My name is Dr. Hamid, and in today's lecture, we are going to study um, exotoxins, endotoxins, their differentiating points, certain bacteria that produces exotoxins, and their mechanism of actions. And then we will also cover very high yield topic, which is hemolytic uremic syndrome for step 1 and step 2. I have started this is microbiology from first aid. I'm making this video because um, this is the topic I was struggling when I was preparing for my step 1. So I thought, why not make some content on basic um, topics that are basic but still hard to understand. Yeah, if you guys have any questions, you can ask me in the comment section. So let's start with exotoxins and endotoxins. What are the differentiating points between these two, which you should remember? First is the source. Exotoxins, from the name, it is very clear they are toxins, endo toxins. Um, Exotoxins, they are present in um, both gram positive and gram negative bacteria mostly, while endotoxins, they are found mostly in the gram negative bacteria and not in gram positive bacteria. What is the composition or chemistry of exotoxins and endotoxins? Exotoxins are made of polypeptide, they are protein in composition, while endotoxins, they are lipopolysaccharide, they have lipid and carbohydrate component. Lipo means lipid, LPS is lipopolysaccharide, lipo is lipid, and polysaccharide is carbohydrate. And lipid A, which is the component of lipopolysaccharide, which is endotoxin. So lipid A is the virulent factor, is the factor that works in endotoxin. You should know. Um, Now, how they are secreted from the cell. Exotoxins, they secrete from the cell. Bacterial cell secrete exotoxins, while endotoxins cannot be secreted. They are only released when bacterial cell undergo lysis. When the bacterial cell breaks down, it releases endotoxins. They Bacterial cell cannot secrete endotoxin. They are only released when the bacterial cell undergo lysis. Okay, this is the image you guys should see. So this is the bacterial cell that is secreting these green are exotoxins. The bacterial cell is secreting exotoxin. This is the bacterial cell that is undergoing lysis, and the lysis of this bacterial cell release endotoxin. So endotoxins are not secreted, they are only released when the bacterial cell undergo lysis. Very important point you guys should remember. Now, the location of genes of exotoxins is mostly in plasmid, which is an extra genetic material in the bacteria. And for endotoxins, it is bacterial chromosome. Um, Exotoxins, they are highly fatal. The toxicity is very high, while endotoxins, they are not very fatal. Clinical features, the exotoxins has various effects, while endotoxins, they usually produces they usually causes fever, shock, and disseminated intravascular coagulation, DIC. um, and how endotoxins causes fever, shock, and DIC. So you should remember that when endotoxin, look at this image, when endotoxins are released by bacterial lysis, they they go and bind to the receptors on the host cell, and then host cell secrete TNF alpha, interleukin 1, and interleukin 6. This TNF alpha, interleukin 1, and interleukin 6, because of endotoxin binding to host cell, um, these three are pro inflammatory cytokines. They induce inflammation, they promote inflammation. So TNF alpha is a pro inflammatory cytokine that causes fever. They cause acute phase reaction, they increase vascular permeability and cause edema. They also mediate septic shock, causes increase vascular permeability. They contribute to septic shock. While interleukin 1 is also a pro-inflammatory cytokine. Interleukin 1 also cause fever. They promote inflammation. Interleukin 6 is also a pro-inflammatory cytokine that causes fever and increase acute phase reactant proteins. So that's how they cause these endotoxins cause fever, shock, because of increased vascular permeability, and they cause DIC, because they induce secretion of TNF alpha, interleukin 1, and interleukin 6. One important point that you guys should know for memorization, that interleukin 1, remember, hot with interleukin 1. So it mostly causes fever. From interleukin 6, always remember sick, and it causes acute phase response, acute phase reaction. And from TNF alpha, tumor necrosis factor alpha, tumor necrosis, so it causes fever, shocking cachexia. So this is how I remember this. Now let's talk about antigenicity, which is very confusing. So what does mean by antigenicity? Here it is saying induces high titer antibodies called antitoxins and endotoxins. They are poorly antigenic. So what does mean by antigenicity? Exotoxins have high antigenicity as compared to endotoxins. Endotoxins have low antigenicity. And what does that mean? Exotoxins have high antigenicity. That means they are strongly recognized by the immune system, by our body's immune system. When the exotoxins are present in our body's immune system strongly recognize them, and our immune system gets activated when they see exotoxins, and our body's immune system start producing neutralizing antibodies called antitoxins to fight against those exotoxins. Okay, as compared to exotoxins, endotoxins, they are poorly antigenic. That means when the endotoxins are present in our body, the body does not recognize them efficiently. They don't The immune system does not get activated as much as they get activated while by seeing exotoxins, and the body does not produce antitoxins against those endotoxins. Okay, so exotoxins, they are highly antigen. They have high antigenicity because the body um, start producing antitoxins against exotoxins as compared to endotoxins. Okay, um, and these exotoxins can also used as vaccines. Um, how they can use it vaccines as and So, exotoxins can be modified to remove their toxic effect, but they keep their antigenic structure. And then we can produce. That's how we can produce vaccines from exotoxins, and those vaccines are called toxoids vaccines. We know that there are three types, I think three or four types of vaccines: live vaccines, inactivated vaccines, toxoid, and then I think subunit vaccine, something. So exotoxins can scientists what scientists can do, scientists can modify these exotoxins and remove their toxic effect, but they keep their antigenic structure, and can form vaccines from exotoxins, and those vaccines are toxoid vaccines. Example are the DTaP, diphtheria, tetanus, acellular pertussis. These are the toxoid vaccines. And as compared to exotoxins, endotoxins, they are poorly antigenic, and they cannot be used as vaccines. We cannot form toxoid vaccines from endotoxins. Okay. Um, Exotoxins, they are rapidly destroyed by 60 degrees centigrade temperature. That means they are heat labile, while endotoxins, they are heat stable. They can um, they they can survive up to 100 degrees centigrade temperature. These are the examples of the diseases which exotoxins and endotoxins can cause. So exotoxins can cause diphtheria, they cause tetanus, botulism, and cholera, while endotoxins can cause meningococcemia, sepsis by gram-negative rods. You should know. Yeah, I think this is all you guys should know about always remember the differentiating points comparison between exotoxins and endotoxins, very important for step 1 exam especially. Now we'll talk about those bacteria that produce exotoxins. So first of all, now you know what are exotoxins, how they are secreted from the body, what is their composition, and um, they are heat labile, and they have high antigenicity. We can use exotoxins to produce vaccines like toxoid, what type of the diseases exotoxins can cause. Now, um, we will discuss bacterias one by one on the basis of their function. On the basis of, look at this. Um, so first, we will talk about those bacteria that produces exotoxins, and these exotoxins, they inhibit protein synthesis. So first, we'll talk about the bacteria that inhibit protein synthesis on the basis of we have categorized bacteria on the basis of their function, I would say. Those bacteria that inhibit protein synthesis, so these are corynebacterium diphtheria, pseudomonas aeruginosa, shigella, and enterohemorrhagic E. coli. So these are four bacteria, and these four bacteria produce different types of exotoxins, but these exotoxins of these all bacteria eventually inhibit protein synthesis in our body. Okay, so first we'll talk about corynebacterium diphtheria and pseudomonas aeruginosa, because they have the same mechanism of action. They have different exotoxins, they produce different exotoxins, and their different exotoxins works similar. So, Corynebacterium diphtheria produces diphtheria toxin, while Pseudomonas aeruginosa produce exotoxin A. But these both toxins, diphtheria toxin and exotoxin A, have same mechanism of action. They have different toxins. They produce different exotoxins, but they have same mechanism of action, and but eventually they produce different type of diseases. Okay, so if that makes sense. I mean, so Corynebacterium diphtheria produce diphtheria toxin and Pseudomonas aeruginosa produce exotoxin A, but they both have same mechanism of action. What is that mechanism of action? They inactivate elongation factor 2. What is elongation factor 2? So usually in our body, what happens is elongation factor 2 is a host cell protein, is a protein that is required for protein synthesis. In eukaryotes, elongation factor 2 is a protein that is required for protein synthesis. It helps ribosome. Okay, it helps. So you know that the cell has ribosome, and it helps ribosome move along the messenger RNA during translation. So elongation factor 2 is required by the ribosome. Um, in the simple words, just remember that elongation factor 2 helps ribosome to move along the messenger RNA during translation. Transcription, translation, these are the steps of protein synthesis. So, just for your convenience, for memorization part, remember that elongation factor 2 is required for protein synthesis. And what happens is, without elongation factor 2, the cells cannot produce protein, they cannot make protein. And when they cannot make protein, their structure, they cannot, eventually they cannot make their structure. And when they cannot make their structure, they will eventually, the cells will eventually die. So what happens is, Corynebacterium diphtheria and Pseudomonas aeruginosa, they produce exotoxins, and these exotoxins inactivate elongation factor 2 via ribosylation. When they inactivate elongation factor 2, elongation factor 2 cannot help ribosomes and ribosome and messenger RNA during protein synthesis, so they cannot make protein, and eventually the cells die. So what happens with Corynebacterium diphtheria? What they cause pharyngitis with pseudomembrane formation in the throat, and they also cause severe lymphadenopathy, which is called bull's neck, and also cause myocarditis. So these two diseases, always remember. First one is very important: pharyngitis with pseudomembrane formation and lymphadenopathy is caused by corynebacterium diphtheria, and pseudomonas aeruginosa causes host cell death. Now Now we will talk about Shigella and enterohemorrhagic E. coli. Shigella and enterohemorrhagic E. coli, easy peasy. They both produce Shiga toxin. Now you should remember just one toxin for Shigella as well as enterohemorrhagic E. coli. They both produce similar toxin, which is Shiga toxin. And what they do is their mechanism of action is also similar. They inactivate 60S ribosome. Ribosome has two units, 60S and 30S, I guess, and they inactivate 60S ribosome. How they inactivate 60S ribosome? By removing adenine from the ribosomal RNA. Ribosome has ribosomal RNA and ribosomal RNA has adenine, so they inactivate 60S ribosome by removing adenine from the ribosomal RNA, and they cause similar disease, which is they cause dysentery. Dysentery means they cause bloody diarrhea. These toxins damages GI mucosa, and then they cause through damaging GI mucosa they cause bloody diarrhea, which is dysentery. Um, now And they also cause enterohemorrhagic E. coli, O157. Enterohemorrhagic E. coli produce Shiga toxin, and um, commonly E. coli, enterohemorrhagic E. coli O157H7, they produce Shiga toxin, and they cause hemolytic uremic syndrome. First they cause dysentery, and then they eventually cause hemolytic uremic syndrome. So now we will talk about hemolytic uremic syndrome. What is hemolytic uremic syndrome? Hemolytic uremic syndrome is a triad of microangiopathic hemolytic anemia, number second thrombocytopenia, and number third acute kidney injury, AKI. Okay, always remember hemolytic uremic syndrome is a triad, three things. Always remember three findings in hemolytic uremic syndrome: microangiopathic hemolytic anemia, thrombocytopenia, and AKI. Okay, and how it happens. So when we eat after, usually it happens after eating undercooked beef or unpasteurized milk or juice or contaminated vegetables that contain that undercooked beef contain they have enterohemorrhagic E. coli, and then enterohemorrhagic E. coli produce Shiga toxin, and then these Shiga toxin, what happens, how they cause hemolytic uremic syndrome. They what they cause, they first damages GI mucosa, and they cause bloody diarrhea, dysentery, and then what they do is Shiga toxin damages the endothelial cells of the kidney. They are damaging, just like they're damaging GI mucosa. In the same way, these Shiga toxin damages endothelial cells of the kidney vessels. So kidney vessels have endothelium, and they damages endothelial cells of the kidney vessels. And when we and then when there is a damage of endothelium, there is activation of clotting pathway. So a platelet, it causes platelet activation, and clotting system get activated and leads to microthrombi formation. When microthrombi is formed in the endothelium, and the RBCs pass through that endothelium, RBCs get sheared as they pass through that narrowed vessels because of the microthrombi formation. RBCs get sheared when they pass through that narrowed vessels and lead to RBCs gets breakdown, and we produce fragmented RBCs, also called schistocytes, and this is how they lead to microangiopathic. They cause hemolytic anemia because of RBCs breakdown. They cause hemolytic anemia. Then the platelets then the body use platelets in the clotting pathway, and the platelets gets used, they get used in that clotting pathway, so that leads to thrombocytopenia. And then kidney vessels got damaged. These kidney vessels get blocked because of the because of what because of that microthrombi, and kidneys does not get enough blood, enough oxygen, so that leads to acute kidney injury. So that's how we call hemolytic uremic syndrome is a triad of microangiopathic hemolytic anemia, and then thrombocytopenia and AKI. So always remember this triad. Always remember what type of E. coli, because there are three four types of E. coli. So always remember remember that enterohemorrhagic E. coli, commonly E. coli O157H7, that produce Shiga toxin, and that causes hemolytic uremic syndrome. Not all E. coli cause hemolytic uremic syndrome. Always remember the pathophysiology. Always remember that it happens after eating undercooked beef or unpasteurized milk. Always remember the lab findings. When the RBCs gets breakdown, it leads to decreased hemoglobin. We'll see in the lab work, in the complete blood count, we'll see low hemoglobin. We'll see haptoglobin gets utilized. We'll see low haptoglobin. We'll see increased level of indirect blue ribbon, not direct blue ribbon. And then, what are the treatment options for hemolytic uremic syndrome? Always remember, we treat hemolytic uremic syndrome. We give supportive care, we give fluids, we give supportive management, and no antibiotics. Antibiotics are contraindicated in hemolytic uremic syndrome, because when we use antibiotics uh, cause increased toxin release, and it worsens hemolytic uremic syndrome. That is why antibiotics are contraindicated in hemolytic uremic syndrome, and we always treat it with supportive care. Okay, now this is a question I want you guys to answer. um, that is So the question is, how we differentiate hemolytic uremic syndrome from thrombotic thrombocytopenic purpura, TTP. How we differentiate HUS from TTP. There are two more findings in TTP which we don't see in hemolytic uremic syndrome. If you guys know, let me know in the comment section. Thank you so much.